Chapter 7, Cell Reproduction, Part 1. Chapter 7, Overview. In this chapter, you will learn about the phases of the cell cycle and how cells divide through mitosis, differences and similarities between sexual and asexual reproduction, how sexual reproduction creates variety in populations, cell cycle control, abnormal cell division, and how cancer can result from unregulated cell division due to a mutation. The cell cycle. The cell cycle is a repeating series of events that includes growth, DNA synthesis, and cell division. Cell division is simpler in prokaryotes than eukaryotes because prokaryotic cells themselves are simpler. Prokaryotic cells have a single circular chromosome, no nucleus, and few other organelles. Eukaryotic cells, in contrast, have multiple chromosomes contained within a nucleus and many other organelles. All of these cell parts must be duplicated and then separated when the cell divides. The eukaryotic cell cycle. The eukaryotic cell cycle has several phases. During interphase, the cell grows, performs routine life processes, and prepares to divide. The three steps of growth 1, G1, which is also known as G1, synthesis, also known as S, and growth 2, also known as G2, are generally grouped together as interphase. The mitotic phase, M, includes both mitosis, which is division of the nucleus, and cytokinesis, which is division of the cytoplasm. In most cells, the cycle is constant. As soon as daughter cells are produced, they too start the cell cycle. I chose this picture to show you. It's a piece of tissue from an onion onion plant stem. It turns out that looking at plant mitosis cell cycle images is more clear than looking at ones from animals, so I provided this one for you, very similar with what would be happening in an animal. And in this piece of tissue here, we can see all of the different phases of the cell cycle. I'm going to move ahead a little bit and have this picture up again as we go through the steps. In growth phase 1, or G1, this is where the cell spends most of its life, during which the cell undergoes rapid growth and performs its routine function. So remember, G1 is within the interphase heading. In here, the biosynthetic and metabolic activities of the cell occur at a high rate. We have the synthesis of amino acids and hundreds of other thousands or millions of proteins that are required by the cell during this phase, including those needed for cell replication. And I'll stop here for a second just to explain here that remember, we're heading towards making two cells out of one cell, basically. So we have to be ready with all of the ingredients for doubling the amount of material to have that second cell. So a lot of growth and biosynthetic activity occurs. So think of replacing, for instance, or doubling the DNA. We have to have the nucleic acids and other components of those, even bases that might be being brought together out of raw materials. There have to be lipids for cell membranes and other enzymes that are important in helping the cell to divide. Now, we should also mention that if the cell is not dividing, and in certain cases this happens, the cell enters something called the G0 step. G0 step is what occurs when we have a type of senescence, a pause in cell division, or maybe just a stop in cell division. So we have cells that maybe have been completely differentiated. They're just going to hang out and maintain their own metabolism, but not increase their number. This is what happens with neurons. If neurons are established and steady, then they will just exist as cells. We do know that neurons can divide in certain circumstances, but they might just stay doing their job in that position. And some cells will stop dividing when issues of sustainability or viability arise. So if there's DNA damage or degradation, then there might be a stop place on division, because then the cell would want to prevent damaged cells to be dividing. And we'll talk about this a little bit later in terms of the checkpoints in the cell cycle. And cellular senescence then also occurs when normal diploid cells lose the ability to divide normally after about 50 cell divisions. So let's talk about that term diploid. Diploid means that the cell has pairs of chromosomes. So in other words, a typical somatic cell, which is a body cell, when you want to differentiate that from a sex cell. So somatic cells are cells in your liver, in your heart, in your eye, and the other type of cell you have in your body is called a germ cell. Germ cells are the cells that exist in the ovaries and testes, or it also refers to eggs and sperm. So there's two different things happening here. But in a normal diploid cell, somatic cells, they have, for instance, 2 chromosomes number 1, 2 chromosomes number 2, 2 chromosomes number 3, all the way up to 2 chromosomes of number 22, and then either an X or a Y, an X and a Y, or 2 X's for the pair of sex chromosomes. And then we do see that cells will reach a level of non-division because their life is over, so to speak. So they're turning over because they've already given rise to many generations of offspring, so to speak. But then perhaps that there'll be some pause in the cell division of the parent cell. This figure here is showing a very simple graphic, but I have it up here in several places because I want you to see the length of the interphase, which we aren't through talking about yet, but interphase is the orange ring. And so you can see if the circle was, let's say it was an hour, then most of that hour is spent in interphase. This is where the work gets done, the products are made to get ready for dividing the nucleus and the cytoplasm. And the other mitotic phase is pretty short in relationship to the interphase. And then within that orange circle, you can see we have G1, S, and G2. And then if necessary, the cell will pause and go into that G0 phase. Okay, now we have the synthesis phase. And this, you could think of the S standing for synthesis of DNA. And you know then for 2 genetically identical daughter cells to be formed, the DNA must be copied through replication. So that's during this phase then, where the amount of DNA is doubled. The doubling of DNA doesn't create a whole new set of chromosomes though. The doubling adds on to the existing set by making a carbon copy of each arm. So the way I like to think about this, let's see if I have a picture of it. Oh, not for a while. The way I like to think about this is if you think about a chromosome as having like an X shape, you might see that in your textbook or other places. Before the synthesis phase, the chromosome wouldn't be an X. It would be just like a capital letter I. And then during the synthesis phase, another capital letter I is made that is identical to the first one. And then if you can picture those coming together into an X, that is what we end up now with after the synthesis phase. Now, the synthesis phase is dealing with DNA that is not very well condensed. It's kind of stringy. It's really in the form of chromatin. Okay, then we have growth phase 2. It's a shortened growth period in which many organelles are reproduced or manufactured. So growth 2 is taking the products from growth 1, the macromolecules, the nucleic acids, of course, have been used in the S phase, and they're taking the next step of modification, the next level of complexity. So we're going from molecules to organelles. And so this is, again, another step getting ready for the division of the cell as a whole. So we have these molecules that are necessary for the division of the nucleus, and other phases of cell division, including these microtubules, which are used in the mitotic spindle, and also phospholipids that are needed to produce additional cell membrane. Okay, and now we're going into now after G1, S, and G2, we're going into the mitotic phase, which is now outside of interphase. Interphase is this orange circle, and then we're going to have the mitotic phase, which is the M. The M in yellow and also the M in purple. The yellow M is the mitotic phase, which is going to be the division of the nucleus plus the division of the cytoplasm here. And the purple M then is the division of the nucleus. The mitotic phase. Okay, so we're going into mitotic now, which is going to be these two steps, mitosis and cytokinesis. At this point, the DNA in the cells, the cell's chromosomes have been replicated. That means the DNA has been doubled and the organelles and molecules have been duplicated. So now the preparation is done, and then we have this next step to actually work toward two separate cells. And we'll see this in mitosis and cytokinesis. So you can look at this greater detail about the mitotic phase in your textbook concept. I want to also at this time encourage you to look at the supportive resources that are available to you for every chapter. If you like to see things move around and have more of a visual experience, then that's where you should go. Have some fun with the stuff that's there because it'll be really useful to you. All right, mitosis is a multi-phase process in which the nucleus of the cell divides. During mitosis, the nuclear envelope breaks down and later reforms. The chromosomes are also sorted and separated to ensure that each daughter cell receives a complete correct set of chromosomes. It has to be the correct set. We don't want too many chromosomes going over into a daughter cell, or not enough. The next is cytokinesis, which occurs in prokaryotic cells. Also, in cytokinesis, the cytoplasm divides and two separate daughter cells form. But in some cells, this cytokinesis does not take place. In some plant species, for example, and in some amphibian species, we have cells that are multinucleated. And also in our human muscle cells, we see multinucleation. Here's a little overview on the cell cycle. We have the resting phase, and interphase includes growth 1, synthesis, and growth 2, and then the cell division itself, resulting in two genetically identical daughter cells. So this is a good point to make, is that through the mitotic cell cycle, what we end up with are genetically identical daughter cells. And that means that the mitotic cell cycle has a particular function. That is for growth and repair. Information you should know. Forms of DNA. Okay. When a eukaryotic cell divides, its nuclear DNA exists as a stringy material called chromatin. Only when a cell is about to divide and its DNA has replicated does DNA condense and coil into the familiar X-shaped form of a chromosome, like the one shown here. So when we're in, let's call it G1 of interphase, the DNA that's in that cell is in this stringy material called chromatin. And even when we go into the S phase of interphase, it's still stringy like this. Well, the stringiness of it is important because if a cell was attempting to replicate DNA that was all bound up very tightly, like a ball of string, enzymes couldn't get inside there to do their job. Remember, DNA polymerase and helicase, etc., have to work on the molecule to, first of all, we need to break the hydrogen bonds and then bring in new nucleic acid molecules. So the relaxed state of the DNA molecule into chromatin allows this DNA replication to occur. And then when we have the process moving ahead further toward actual division of the nucleus, then the chromosomes become condensed and coiled. And you can imagine that this is a better way to transport chromosomes. If you wanted to have part of the chromosomes go into what will be the new cell, and the other part of the chromosomes go into the part that's the other new cell, then they have to be manipulated. If they were all stringy at that point, it would be very, intense to get everything to go where it needed to go. And remember, these are being controlled by the cytoskeleton elements and enzymes. So in this time then, when the DNA has been condensed again, it now is existing in two identical copies. So this is where we have that capital letter I before S in interphase. And then after S, because the cell has duplicated its DNA, we have basically two letter I's that are carbon copies of each other. And if you could picture that now becoming this X shape. So each side of this molecule is an entire copy of that DNA. Each side of it is called a sister chromatid. The sister chromatids are joined together at a region called a centromere here. Okay, so now we have basically one big structure, so to speak, that has double the original DNA material. Okay, and then we're going to have a division of the nucleus in this stage called mitosis. And mitosis now includes different steps also. So mitosis includes the steps of prophase, metaphase, anaphase, and telophase. And in these steps, we're going to come from that condensed kind of double chromosome structure into two cells in which the DNA material is identical. Okay. And here's a picture that we can look at in general to see how this happens. We can look at the interphase step here where we're not seeing chromosomes because they are uncondensed. They're that stringy chromatin. And then in the prophase of the mitotic phase, the chromosomes are now have been condensed. They started a little bit at the end of interphase. You could see them in a microscope at this stage. And then as we go, they get more condensed. They start to be rearranged because the nuclear envelope breaks down and allows them to be manipulated. Again, we're talking about the centrioles and the spindle and enzymes that are moving these chromosomes around. In metaphase, they line up along what's called the equator. In anaphase, they start to be pulled apart. And what happens is that they pop apart at the centromere. We'll go into some detail on this. So that the double chromosomes now are single chromosomes that are pulled to opposite ends of the poles of the cell. And then they move farther to the poles and a new area of nuclear envelope forms separating them. Finally, at telophase, we have two separate cells that now are going to probably go right straight through interphase again. All right, so we'll look at these. Let's see if I put those. Yeah, I did put those pictures in. This is the longest phase of mitosis. In this phase, the chromatin condenses into chromosomes. The nuclear envelope breaks down, And in animal cells, the centrioles, which are near the nucleus, begin to separate and move to opposite poles. Remember that these centrioles are small organelles found only in eukaryotic animal cells that help ensure the new cells that form after cell division. Each will contain a complete set of chromosomes. So this is the thing where we're not going to look for in this example down here. This is a very simplified example. In humans, we would have 23 pairs of chromosomes. In here we have, looks like we have about 2 pairs of chromosomes. And what we're going to have is a situation of equality that's desired. We don't want to have 4 chromosomes in one new cell and no chromosomes in the other cell. So it is orchestrated by these proteins. All right, now here, let's see if we can find a prophase cell. A prophase cell would look kind of like this one here. You can see the nuclear envelope is kind of breaking apart on it. You can compare that to other cells where it looks like the nuclear envelope is very much intact, like this. So that might be a prophase cell right there. In metaphase, then, the spindle fibers attach to the centromere of each pair, and they line up at the equator. So you can think of metaphase as being middle. The spindle fibers ensure that the sister chromatids will separate and go into different daughter cells. Some spindles do not attach to the kinetochore portion of the centromeres. These spindles are called non-kinetochore spindles that help in the elongation of the cell. Okay, so I guess we could talk about the elongation right here, and you can see how the cell is getting bulgier. These are the non-kinetochore spindles here that are pulling against the membrane, elongating that. Remember, this is going to be 2 new cells. You can see the organelles inside here have also been distributed so that there's Golgi and mitochondria and lysosomes and everything else. Smooth ER, rough ER, all needs to be separated. And that these chromosomes now that are lined up are going to separate. And let's see how that happens. Let's see if we can find a picture of that here. We have metaphase. We could find metaphase here in this onion cell, and it looks kind of like eyelashes along the equator of the cell here. That's metaphase. And we can look at that. They're lined up, and then we can see what's going to happen next in anaphase. And so in anaphase, the sister chromosomes are pulled apart by the shortening of the spindle fibers. So it's like a tension that's on these double chromosomes to pull apart at the centromere, producing now 2 sets of single chromosomes, if you can think about it that way. This pulling apart is a little like reeling in a fish by shortening the fishing line. In this case, though, we have like the fish is going to split in half, and half's going to go in one direction, the other half goes in the other direction. One sister chromatid moves to one pole of the cell, and the other sister chromatid moves to the opposite pole. At the end of anaphase, each pole of the cell has a complete set of chromosomes. There are 2 sets of unreplicated chromosomes in one cell. Let's see if we can find that picture. They're being pulled apart, maybe like this one here, and maybe like this one here. They're being pulled apart. Telophase. In telophase, the chromosomes reach the opposite poles and begin to decondense into a stretched out chromatin configuration. So they're going to relax again, likely going back through interphase where that chromatin form of DNA can be replicated. The mitotic spindles are also depolymerized, which means that they relax into smaller components, and that these now tubulin monomers are used now to assemble some cytoskeleton components for the daughter cells. So they're being recycled from one use to another. Nuclear envelopes form around the chromosomes, and nucleosomes appear within the nuclear area. Nucleosomes consist of about 150 base pairs of DNA wrapped around a central core of 8 histone proteins. So we could go back into our chemistry chapter to look at the structure of DNA as it coils around itself, and creates this beads on a string structure that actually organizes this DNA so that it can be packaged and protected within the cell. Telophase then is going to be a situation here where the chromosomes are mostly pulled to opposite poles, and even in this one here you can see the cell membrane is starting to form to separate the 2 new cells. Next, we have cytokinesis, the final stage of cell division in eukaryotes as well as prokaryotes. During cytokinesis, the cytoplasm splits in 2 and the cell divides. The process is different in plant and animal cells. In animal cells, the plasma membrane of the parent cell pinches in along the cell's equator until 2 daughter cells form. This pinching in is a result of contractile proteins that are in the cytoplasm, another function of proteins. In the plant cells, a cell plate forms along the equator of the parent cell. The cell plate is often where the cellulose in the plant is present. So this is the stiffener that goes into plant cells that makes them different than animal cells, so that a plant stem can stay upright and not just flop over. That's due to the cellulose in the cell plate. Then a new plasma membrane and cell wall form along each side of the plate. And here is another picture of that onion cell, and cytokinesis then can be observed in here. Kind of, again, these ones that are kind of looking like they're split in 2. I don't see one that's exactly just separating or not, but you could have some fun looking at these particular slides to see if you can pick out all the phases of mitosis.